Okay, so welcome to this first video in the playlist on lipid metabolism. In this video, what we're going to do is talk about the different types of lipids. Okay, so this is going to be a basic introduction to lipid molecules. Okay, right. So we're going to start off with the different types of lipid. We're going to talk about the 4 different types of lipid molecule. There are others that don't fit into any of these 4 categories, but they are the minutiae ones. So we're going to talk about the major 4 categories of lipid molecules. We will then talk about their uses, okay, and then we'll go through each type and have a look at the structure of them. Okay, right. So let's start off with the different types of lipid molecules. So by far the most important of all of the lipid molecules are what are known as neutral fat. Okay, so neutral fats. And the other name for neutral fat molecules, probably the more common name for a neutral fat molecule, is to call it a triglyceride. Okay, so triglycerides are the main forms of lipid molecules. Okay, now there is another name for triglycerides. Triglycerides, strictly speaking, is short for triacyl glycerols. Okay, right. And for short, triacyl glycerols can be abbreviated to, whoops, not like that. How can I rescue this? I'll sort of do a T on the slant like that. TAGs. Okay, so T for tri, A for acyl, and G for glycerols. Okay, so triacyl glycerols, triglycerides, neutral fat molecules, whatever you want to call them. Okay, they are the most common form of lipid molecules, and we'll discuss their structure in a moment. Okay, the second type are what are known as phosphoglycerolipids. Okay, so phosphoglycerolipids. And when I probably say phospholipid, phosphoglycerolipids is probably what comes into your mind, okay? So phospholipid is a more broad word, as we'll see, okay? Phospholipid does refer to phosphoglycerolipids. All phosphoglycerolipids are phospholipids, but phospholipids is broader, basically. There are other types of lipid molecules which do not fall into the category of phosphoglycerolipids, which do fall into the category of phospholipid. Okay, so be wary when you use the term phospholipid because phospholipid does refer to phosphoglycerolipids, but they're probably what you mean and not the other forms of phospholipids. Okay, right. So the third form of lipid molecules is what's known as sphingolipids. Now, some of the sphingolipids are actually phospholipids. Not all sphingolipids are phospholipids, but some of them will be. So in fact, the term phospholipid refers to all of the phosphoglycerolipids as well as some of the sphingolipids. Okay, and we'll look at that in more detail when we look at the structures of these molecules. Okay, and then the final category of lipid molecules is going to be cholesterol. Okay, now you might be wondering, well, what do all lipid molecules share? Why are they all sort of clustered together as lipids? Well, basically, the sort of characteristic feature that all lipid molecules have is that they're very hydrophobic molecules. Okay, so the structures are not particularly polar. There's a lot of neutral molecules, basically. They're very neutral, and that makes them hydrophobic. It means that they can interact well with other neutral molecules, other non-polar molecules, but they don't interact well, for instance, with water, which is the reason they're called hydrophobic. And the reason is that water is a very polar molecule, okay? So water can form very strong interactions with other polar molecules, such as other water molecules, okay? And therefore, it would not be a good idea for water molecules to interact with these hydrophobic molecules, because the interactions that you'll have between the hydrophobic molecule and the water will be much weaker than the interactions that you have between 2 water molecules. So basically, it's just not thermodynamically viable for 2 water molecules to split apart their intermolecular bonding in order for a water molecule to instead bind to a hydrophobic molecule, because the interactions which it will form with a hydrophobic molecule are just far weaker than the interactions that it can form between itself and another water molecule. Okay, right. So, what are the uses of lipids then? There are two major uses of lipids. Firstly, we can break them down and convert them into energy, basically. So metabolism is one of the uses of lipids. So we can break them down and use the energy that we get from the catabolism of the lipid molecules to produce ATP, adenosine triphosphate molecules. And for that purpose, lipids actually sit alongside carbohydrates, okay? A lot of your metabolism is lipids, just as a lot of your metabolism is glucose. It's around 50-50, so lipids are very, very important for metabolism. Okay, right. The other use of lipid molecules is in the building of lipid bilayers, okay, or membranes. So basically, we know that all cells in the human body have a cell membrane surrounding them. Okay, this is a lipid bilayer. In addition, the nuclei of the cell, or the nucleus rather of the cell, will have a lipid bilayer surrounding it. Okay, you also have a whole bunch of intracellular organelles, such as the endoplasmic reticulum, the Golgi apparatus, these sort of things. They are all membrane-bound organelles, and they have one of these lipid bilayers surrounding them, basically. So, what are these lipid bilayers actually constructed of? Well, they're constructed of lipids. So basically, you've got two layers of lipids. So let me show this. So this is how people usually draw a lipid bilayer. And basically, you've got one layer, which I'm showing in turquoise here. Okay, and then underneath that, you've got another layer, which I'll show in pink here. Okay, right. So these two layers of lipids sit on top of one another. They're two surfaces of lipids. They sit on top of each other. The hydrophobic portions from each of them interact very nicely with one another, and then they often have polar groups which face out into either the extracellular fluid or the cytoplasm, okay? So those will face out towards the water. So often what you'll have is that the lipid molecules composing these layers out of which we've constructed the lipid bilayer will have polar groups which face outwards, basically. So you'll have polar groups facing out in either direction. So this side will have polar groups as well, and those will interact nicely with the water that the two sides of the lipid bilayer are in contact with. Okay, right. So let's now start talking about the actual structure of each of these four types of lipid molecules. So we'll start with the structure of a triglyceride or a triacyl glycerol. Okay, right. So triacyl glycerol is actually the better name for this because it tells you exactly what we're talking about here. It tells us that we're dealing with a glycerol molecule which has had 3 acyl groups added onto it. So let's start off with the structure of glycerol. So basically, the chemist's name for glycerol is propane 1,2,3-triol. Glycerol is kind of the biochemist's name for it. The hardcore organic chemist will call it propane 1,2,3-triol. Okay, and although propane 1,2,3-triol is a bit more of a mouthful than glycerol, it's a useful name because it tells you exactly what we're dealing with here. It tells us that we're dealing with a 3-carbon molecule where we have alcohol groups coming off the first, the second, and the third carbons of that molecule. Okay, so let's draw a molecular structure for this. So we've got 3 carbons, like so. We then have an alcohol group coming off the first carbon, so let's label this one up as the first. Okay, let's label this one up as the second and this one up as the third. We've then got an alcohol group coming off the second carbon and an alcohol group coming off the third carbon. And then to saturate these carbons up, just stick a hydrogen off wherever you have a free bond. Okay, like so. So here is our propane 1,2,3-triol molecule. Okay, right. So what's important about this propane 1,2,3-triol molecule is that you have these 3 alcohol groups, one coming off each of the 3 carbons, okay? And to turn a propane 1,2,3-triol molecule or a glycerol molecule into a triacyl glycerol molecule, what you need to do is bind acyl groups onto these alcohol groups. So what is an acyl group? Okay, right. So in order to understand what an acyl group is, ACYL, we need to understand something about carboxylic acids. So let me just draw an arbitrary carboxylic acid here. Okay, so this R here denotes that you can have an arbitrary group there. Okay, so here's the carboxylic acid group here. Basically, hopefully you know that carboxylic acids can react with alcohol groups, okay, to form what are known as ester links. So basically the reaction consists of taking the alcohol group off the carboxylic acid group, taking the hydrogen off the alcohol group, binding those two together to produce water, okay, and then binding the carbon of the carboxylic acid molecule to the oxygen of the alcohol group to create what's known as an ester link. So let me draw this out again. So here's our carboxylic acid molecule now linked via this ester link to the alcohol group. Okay, so this is called an ester link here. So basically, you can react free carboxylic acid molecules onto a glycerol molecule, and this is in fact what we're going to do. Now, why do you not call this a tricarboxylic acid glycerol? Okay, what does the acyl mean? Well, what you'll notice is that the actual portion of the carboxylic acid molecule that we add on to the alcohol group is not the entire carboxylic acid molecule. Instead, we only add on this portion here, which I'm now circling in green. Okay, this portion without the alcohol group that's usually on the carboxylic acid group is then called an acyl group. Okay, or the acyl group of a carboxylic acid. Okay, right. So that is why this is called, that's why what we're about to draw is called a triacyl glycerol, because actually you don't add the full carboxylic acid molecule on. When we esterify free carboxylic acid molecules onto these free alcohol groups, we're not going to add on free carboxylic acid molecules, we're only going to add on the acyl groups of those free carboxylic acid molecules. Hence why the final molecule is going to be called a triacyl, free acyl groups on glycerol, okay, or a triacyl glycerol, a tag molecule. Okay, so let's now draw a triacyl glycerol molecule then. Okay, now the other thing I need to tell you before we can actually go and draw a triacyl glycerol molecule is that the carboxylic acids we are going to esterify onto these free alcohol groups of the glycerol molecule are going to be extremely long carboxylic acids, i.e. this arbitrary group here is going to be a very, very, very long hydrophobic tail, basically. So I need to discuss the main three long-chain carboxylic acids that we usually esterify onto a glycerol molecule to produce a triacyl glycerol. Okay, right. So we're now going to discuss what are known as long-chain carboxylic acids. Okay. However, long-chain carboxylic acid is once again the organic chemist name. So this time we want to go from the organic chemist name to the biochemist name. The biochemist name for a long-chain carboxylic acid is to call it a fatty acid. Okay, so let me give you some common examples of long-chain carboxylic acids which can be esterified to the alcohol groups of glycerol molecules to produce triacyl glycerol molecules. Okay, so the first example is stearic acid or stearic acid. Okay, now stearic acid is the biochemist's name. Okay, the organic chemist name for stearic acid is to call it octadecanoic acid. Okay, and once again, the organic chemist's name is more useful than the biochemist's name because octadecanoic acid tells us immediately what we're dealing with here. It tells us that we're dealing with an 18-carbon, fully saturated carboxylic acid. So here's our carboxylic acid group. Now we need to take it up to 18 carbons long. So we've got one here. We now need to draw the other 17. So 16 of them are going to be in the form of methylene groups in the middle. So here's the methylene group, and now I don't want to have to draw 16 methylene groups out. Besides, I don't actually have the space. So there's a clever trick. What you can do is circle one methylene group which you have drawn, and then, well, not circle it, bracket it, okay, and then suffix that bracket with a 16, okay, so this means you've got a carboxylic acid group at the end, then you've got 16 methylene groups following that, and then right on the end what you have is a methyl group like so. Okay, and that takes you up to 18 carbons because we've got 1, then we've got 16, which takes us up to 17, and then the 18th there, that's octadecanoic acid, and as you can see, hopefully, it's fully saturated. Okay, so that's an example of a long-chain carboxylic acid, or a fatty acid. Okay, right, that's not the only example, though. I'll now give you another example. Okay, so I'm going to tell you about the 3 main long-chain carboxylic acids. We've seen stearic acid. Next one we're going to look at is oleic acid, and then we'll go to palmitic acid. Okay, so oleic acid next. Right, so oleic acid is once again a 18 carbon carboxylic acid, but this time it's not fully saturated. Okay, so if you want the proper chemist name for this, it will be and this time it's not decanoic acid, it's decanoic acid. But then we have to tell you what carbon the double bond is coming off. So what you do is you put octadecene, that shows us that you've got a double bond at some point. Then you put a 9, and then it's decanoic acid, 9-enoic acid. So people would call this octadecanoic acid. Okay, right. Let's draw the structure of this then. So, here is our carboxylic acid group. Now, the double bond is going to be between the 9th and the 10th carbon. It's going to be exactly in the middle of the molecule, which is 18 carbons in total. So, there are going to be 9 carbons on this side of the double bond, and 9 carbons on this side of the double bond. So, we're firstly going to have 7 methylene groups. Okay, again, I don't want to have to draw out 7 methylene groups, so I'm just drawing 1 and then bracketing it and putting as the suffix 7. Okay, then here is now the 9th carbon, so here's carbon 1, then this takes us up to carbon 8, here's carbon 9, where the double bond is going to come off. Okay, so here's carbon 9, here's carbon 10. Okay, let's put hydrogens coming off them. Okay, then of carbon 10 we're going to have methylene groups. Okay, so we now have 8 more carbons to do. 7 of those are again going to be as methylene groups, so I'll just draw 1 methylene group, bracket it and put a 7 there, and then the final one is then going to exist as a methyl group right at the end. Okay, so this is the structure of octadecanoic acid. Okay, so let's just check. We've got 18 carbons in total. We've got the first one here, then we've got 7, that takes us up to 8. The 9th one's here, 10. Then we've got another 7, that takes us up to 17, and then 18 here. So that's oleic acid. Okay, the final long-chain carboxylic acid, which is very commonly used within triglycerides, is palmitic acid. Okay, and this is another nice, easy one. Okay, so the proper chemist name for palmitic acid is to call it Hexa decanoic acid. And once again, this is a better name because it tells... Whoops, I've missed off the C. I can turn that into an A, though. There we go. Octadecanoic acid. Sorry, not octadecanoic acid, hexadecanoic acid. So this is a 16-carbon, fully saturated carboxylic acid. Okay, so here's the carboxylic acid group. Now we need to show the other 15 carbons. So we're going to have 14 methylene groups. Okay, so we'll draw 1 methylene group. We'll bracket it and subscript it with a 14 here, and then we'll have 1 methyl group right at the end, and that takes us up to 16 carbons. So that's the structure of palmitic acid. Okay, right, so those are the structures of the long-chain carboxylic acids. So, going back to the structure of a triacyl glycerol, what we're going to now do is esterify 3 long-chain carboxylic acids to these 3 alcohol groups. Now, Obviously, there are many different long-chain carboxylic acids that you could put in each of these positions. So there is not just one triacyl glycerol molecule. There's a huge number of different molecules which would all be considered triacyl glycerol, because you can have different long-chain carboxylic acids esterified to these free alcohol groups. Generally, they are either steric, palmitic, or oleic acid, but there are others as well. Okay, so I will give you an example of one triacyl glycerol, okay, which is the one where you have free steric acid molecules bound to the free alcohol groups. Okay, so here is our glycerol molecule, like so. So I'm not showing the alcohol groups yet, because those are now going to be involved in ester links. Okay, so here are the oxygens of the alcohol groups, but remember, when we form an ester link, we take the hydrogen off, so that shouldn't be there anymore, and instead this oxygen should now be linked to the carbon of the carboxylic acid group of the carboxylic acid molecule. Okay, right, so here are the carbons of the carboxylic acid molecules, okay, and I'm drawing the triacyl glycerol, which corresponds to having free steric acid molecules bound to the free alcohol group. So one steric acid molecule to each alcohol group. So here is the carbonyl group of these carboxylic acid carbons. Okay, and now we need the R group of steric acid. Now, basically, here I drew you out the methylene group. Exactly, I showed you the carbon with the two hydrogens. Because there's not much space here, and I think otherwise I'm not going to be able to fit it in, what I'm going to do is start just denoting that methylene group as CH2, okay, rather than actually drawing it physically out. So what I'll do now is I'll put CH2 here for a methylene group, and we're doing steric acid, so we want 16 of those, and then right on the end we want a methyl group, and again I'll denote the methyl group just by H3C rather than drawing it completely out. That's just because I haven't got really room to draw them out as far as the vertical directions go. Okay, so here's another steric acid molecule, so now I just need to copy out what I've just drawn here. Okay, so here are my 18 carbon fully saturated carboxylic acids bound to this glycerol molecule. Okay, and finally another one here. So this triacyl glycerol molecule would specifically be called tristerin, basically, okay, because it's got free steric acid molecules bound to the free alcohol groups of the glycerol molecule. So the specific name for that triacyl glycerol is tristerin. Okay, right, so we'll call it there for this video, and in the next video what we'll do is begin our discussion of the phosphoglycerolipids.